guess we'll go ahead and get started. Hello, everyone. Welcome to GRIN 101. In this session, we're going to go over a series of short talks to discuss some of the basics of GRIN science and the path to cures and therapies. My name is Meagan Collins, and I'm the research coordinator for Cure GRIN. I'm going to be the moderator for this session. So if you have any questions throughout the session, please put them under the Q&A, and we'll try to answer them after our three speakers have presented. And first up, we have Genetics 101, which will be presented by Eriskay, Liston, who is a genetic counselor working in the Division of Clinical and Metabolic Genetics at the Hospital for Sick Children.
Great. So I'm hoping everyone can hear me now. It's a real pleasure to be here today. I wanted to thank Keith and the team for inviting me to speak. I've had the great privilege of working with Keith and his family at Sick Kids. So again, thank you, and very excited to be here. So today we have a brief talk on sort of Genetics 101. I'm going to highlight some key sort of basics about genetics. And I wanted to take a little bit of time going over what a genetic report looks like and how to understand it. And then at the end of the session, we'll have some time to answer some of your questions. So this picture here may bring you back to high school biology. This is a picture of our chromosomes. So if you peered inside any one cell in your body and looked inside the nucleus, you would see this set of chromosomes. And what's important to see here is that our chromosomes come in pairs, and we have 23 pairs. And each chromosome or set, because there's two, you have one that has come or been inherited from your mother and one from your father. So again, as a total of 46. And it's really critical to have that number and that each chromosome is structured correctly. So if there's any errors or differences in the number of chromosomes or the shape or structure, it can result in differences in how your body functions and works. So if we look a little bit closer and kind of unwind that chromosome and take a deeper dive, you can see that each chromosome is made up of thousands of genes. And we think today that there's probably about 20,000 genes in total in the human body. Each gene is actually made up of these structures called base pairs. And amazingly, there's actually only four. There's adenine, thymine, guanine, and cytosine. So A, T, G, C. And incredible to think that just these four base pairs give us such diversity in our genetic makeup. And it's the combination and the unique combination of these four base pairs that lead to the production of a protein in our bodies, which then goes off into the body to perform an important function. And any differences in these four base pairs, or what we call spelling errors or variations or mutations, can cause these proteins to be formed differently or sometimes not at all. So a protein, for example, in the case with GRIN, you can see that the GRIN1 gene, for example, codes for a protein called the GluN1. And these proteins are critical in coming together, or subunits coming together, to activate a receptor in the brain, which then allows for appropriate sort of neurotransmission or communication amongst the brain cells. So anytime there's a difference or change in one of the genes that codes for this protein, it can cause this receptor to not act properly. So what does a variation look like? So this is something called a missense mutation, and this is, or variant, and this is one of the most common types of genetic changes that we can see in the case of the GRIN genes. So this here is sort of a string or pattern of those base pairs that we saw previously. And you can see that together, when there's three in a group, they code for something called an amino acid, which is like a chain or a link in the chain that goes to make the protein. So here you can see there's a repetitive sequence of the CAT over and over again. And just one little change, where there should be an A, there's now a C, that has changed that amino acid and can change the complete ultimate production of a protein or function of a protein. So how do these variants or genetic changes get passed on? So there's different types of inheritance patterns, and I don't have time today to get into all the details, but the most common ones that we see and have been seen in GRIN are called autosomal dominant, meaning that you have one parent has one genetic change on one copy of their chromosome that is enough to cause the condition in that individual. So in this picture, you can see that the father has a genetic change on one of his chromosomes, and the other copy of his chromosome is unaffected or unchanged. While the mother, both copies of that, let's call it GRIN gene or GRIN1 gene, are unaltered or unchanged. And when they go on to have children, it's going to be a 50-50 chance of which chromosome the father will pass on. Will he pass on the copy of the chromosome with the variant or change causing the GRIN1 disorder, or the one without? And what that ultimately results in is a 50-50 chance of their children to have the condition. In an autosomal recessive type of inheritance pattern, what you see is, in fact, that having one copy on one of the chromosomes with the genetic variant, in fact, is not enough to cause the condition. And it's only when both parents, both the mother and father, carry a variant on one copy of their chromosome, and when they both pass that changed copy down to their child, that it can cause the condition. And that what ends up happening there is there's a 25% chance of their offspring having this condition. Now, we know that most of the cases of children with GRIN1 or many of the GRIN disorders are the result of what's something called a de novo variant change, meaning that it's new in that child. So what happens there is that variant happens new in either the egg or the sperm when going to be passed on to the child. So neither of the parents actually carry it in the rest of their body. It just happens sporadically in that egg or sperm that went on to make that child. And then that child will have that copy of that variant in every single cell of their body. And then it would be, again, if that child goes on to have children, would be a 50-50 chance of whether it gets passed on to their children. So just skipping forward to talk a little bit about genetic testing and how do we look for these variants in these genes. So nowadays, most people may have heard of exome sequencing. This is a technology or a test that's happened probably in the last 5 years. And instead of looking at one gene at a time, like looking for a GRIN1 or GRIN2 gene, it looks at all of our genes in one go and basically assesses all of the potential variants in that gene, and then tries to understand, whether that variant is causing the condition or not. So exome sequencing has really increased the rate of diagnosis of many different conditions. And at Sick Kids, we found that probably about 25 to 40% of children are able to have a diagnosis with exome sequencing. And the type of result that you might get, now Keith's family very kindly allowed me to share this result of their child. But what's really important to see here, because we don't have a lot of time, but what I want to highlight for people who are going through genetic testing, first of all, really important that this type of a test is done with a specialist who understands how to order the test and also how to interpret the information and relay that information back to the family. But secondly, to understand what type of a variant the lab has found. So we know that variants we all have variants, we all have genetic variants, but what's important to understand is whether the variant is actually damaging enough so that the protein is not working anymore and what we call pathogenic, or if that variant is just a benign change and actually is a change but not really causing any impact on the actual function of the gene. So what you really want to look for is what type of variant was found, what gene was it found in. So here you can see the gene here was GRIN1. And then also the lab will include evidence about how they've classified that. So the important piece here is that as we're doing more and more testing, our evidence and our understanding of these variants in these genes is growing and changing. And so sometimes, you know, maybe somebody's gone through this testing 4 or 5 years ago. We now have so much more information and data to be able to interpret and explain that finding. And so it can become sort of like a living, breathing document that can change over time, and why it's so important to have a specialist interpret that information. And so basically what I was saying before is that these variants can be classified as highly damaging or benign. And again, very important to understand that these can change over time with new evidence as we do more and more testing for these types of variants. So I think that's all of our time today. Thank you very much again for your attention. I know we covered a lot of information, a lot of details, so hopefully we'll have time at the end to answer some of your questions.
Perfect. Thank you so much for that, Eriskay. Next up, we have The Path to a Cure, which is going to be presented by Dr. Angie Serrano, who's a postdoctoral fellow at the University of Utah, working on disease modeling and developmental biology, and who is serving as our CZI Rare as One scientific advisor.
Sorry, can you hear me now? Sorry, I was mute. Okay. So 1 second. Okay. Well, thank you so much to Meagan and Keith for having me here in this really exciting session. And thank you to all of you for opening the doors of your home, because I feel that somehow by coming all together here, we are meeting you, knowing you, and entering into your homes. So the idea of this talk is to kind of walk together through what we consider is the path to cure, but most importantly, what's our role as family and as patients into this path to cure. So before going to that, let's see some numbers in rare disease. So in the United States, we define a rare disease when it affects fewer than 200,000 people. However, if we put together all the rare diseases that we know to this day, that is around 7,000 different rare diseases, we will understand that 1 in 10 Americans are affected by some kind of rare disease. And that's a huge number. And it should be enough to kind of motivate the development of therapies and incentivize cure. However, we know, and this is very sad, that approximately 95% of them do not have any kind of treatment option. However, in 1983, the FDA created this Orphan Drug Act that will help to give incentive and motivation to pharmaceuticals and research groups in order to develop, push the development of drugs and therapies. So before this act was in place, only 10 new drugs were approved by FDA in 10 years. After this, 770 drugs were approved. So that was a really good step forward towards this. However, we are still facing some challenges, and this is very inherent of what a rare disease is. One of the main points that we need to kind of work around is that we do not have a lot of knowledge about the history and progression of this disease. This could be because we just discovered, we just know about the gene that is causing it, or simply because we are not organized in order to gather all this history and progression of this of the rare disease. Something that it was mentioned previously by Eriskay, and for also Ben mentioned something about this in the previous session, that we have a lot of differences in patients. The heterogeneity is huge. And we can also attribute that to specific genetic subsets, where the mutation is, how is it affecting, is it giving us function or losing function? So all those things will contribute to another challenge, which is a variety of different clinical manifestations. Also, we don't have sometimes a precedent of drug development. That makes it a bit harder. Currently, it's working very well repurposing a drug, something that has been already approved for another disease. We use and we assess if it goes together with the biology of, let's say, GRIN, we can try to investigate that specific drug and repurpose it. And that sometimes goes a bit faster. There is a very limited number of patients for trial. It's very hard already to organize and to motivate patients to be consistent in a trial. If on top of that we have a very small population, that's even harder. And another issue that it was also brought up in the previous session is that 50% of rare disease patients are children. And that also brings some ethical concerns. But here the big question is, how do we translate all of this into cure and therapy for our rare disease? And most importantly, how can you help? How can I help? How can we push together a bit faster? So what I'm going to walk you through in the next 3 slides, it's about what I see the path to cure. And this is also based on Dr. Feigenbaum's view of how we should approach these kind of problems. And this is based in a centric vision of the patients and the families. So in this case, patients and family are going to be the motor, the engine that is going to be moving forward the whole circle of this path. We first need to learn research. We need to learn about this disease, the biology, understand the genes and how this is affected, understand the cell types that are affected, the tissues that are affected, and all this comes from a very deep literature search and research. We also need to develop a research organism and system development that will allow us to then test our hypothesis. So first, research. Once we're at that stage, we can move to preclinical assays. So we use some of the models that we developed before, the knowledge that we gathered before, and we start with some clinical assays, preclinical assays that will allow us to test hypotheses to see if which pathway or which biological point is more affected than the other one. Then we have the very famous clinical development. This is probably extremely long. I haven't mentioned it before, but research is 3 to 4 years. Preclinical assays are 4 to 5 years approximately. And clinical development is already long. In rare disease, it's extended by 3 years. So it could be from 4 to 7 years. This is just so we have kind of a sense of how much it takes to do this in a safe way. So in clinical development, I'm not going to go into detail in the different phases, but we have the phase 1 is basically to test the safety of this drug in a very small healthy population, 20 to 80 individuals. The phase 2 will test the effectiveness of the dosage and method of administration. That is performed in phase 2 already in patients that are with that disease. And in phase 3, we test the efficacy and safety in a bigger population that should be around the thousands. So this is basically what the path that we have to push a drug or a compound into clinical development. And lastly, but not least important, is the awareness. How important is this like in motivating campaigns and making not only our families and local communities aware of the path that we have for diagnosis and for cure, but also healthcare providers, med students, researchers, pharma industries, and of course foundations and donations in order to be able to gather funds. And as I mentioned before, the most important and key player in this cartoon that I'm presenting to you today is patients and families, because there is a direct synergy and communication for researchers to learn from you, from the things that you as a family and as a patient need, so we can ask the right questions in the right moment, and also to be able to push towards preclinical and clinical development all the rest of this path of cure. So basically, that's what I wanted to share with you. The fonts are a bit small in the details, but I'm going to be super happy to answer any questions. But before finishing, I said thank you for opening the doors of your home, and I want to do the same thing. This is my family from Argentina, and I believe that we start with the first step, increasing awareness, and what my family wanted to be here together with all of you in this virtual conference. So thank you so much, and I will be super happy to answer any question at the end of the session.
Perfect. Thank you so much, Dr. Serrano. Now we will learn about GRIN pharmacology, which is being presented by Katheron Intson, who's a PhD candidate working in Dr. Amy Ramsey's lab in the Department of Pharmacology and Toxicology at the University of Toronto.
Hi, everybody. It's a great pleasure for me to be here today and talking to all of you. I'm always really inspired by these conferences and chatting with parents. So as Meagan said, my name is Kath, and I'm a PhD candidate in Dr. Ramsey's lab. And today I'm going to be talking to you guys about GRIN pharmacology. And I know that pharmacology is kind of this big and scary word, and it's not supposed to be a science talk. So the goal of this talk is to make GRIN science moderately accessible to the parents while only moderately upsetting the science by cutting out those details that are often so important to us. One of the most common questions that I get from parents is, why have we decided to group all the kids genetically like this when all of them look really different? Is this even an important classification? And so the goal of my talk today is to tell you a little bit about why the kids are all so different. And so if you have a light bulb moment during this talk, that's what I want you to have is to understand why the kids are so different. Okay, there we go. So many of you parents, especially if you're not newly diagnosed, will have seen a map like this before. There's a scary looking protein, and then there, in order to provide information about what your child's variant is, there's just a bunch of numbers on a screen and, maybe a dot in the center of this like spirally mess that's supposed to tell you something about your child's variant. And this can be really inaccessible and confusing. So let's kind of break it down a little bit. If you're not aware, the GRIN genes encode for proteins that form this complex called the NMDA receptor. And the main job of the NMDA receptor is to sit in the membrane of cells and very broadly speaking, let calcium and sodium channel ions into the cell via a channel and let potassium ions out. And of course, it's a little bit more complicated than that. A magnesium ion sits in the channel and acts as like a gating mechanism to gate whether ions are allowed into and out of the cell or not. And on the outside of the cell, we have these little parts of the receptor that stick out. And, we require glycine or D-serine and glutamate to actually be bound to this receptor in order for the receptor to work at all. And there's also all of these other binding sites, many of which are being discovered newly each day, which allow for modulation of this receptor, so to let more ions in or fewer ions in. And so it takes 4 of these proteins that are encoded by the GRIN genes in order to form this actual functioning receptor. So I just wanted to simplify the life's work of Dr. Trainellis and Dr. Furukawa into one slide and kind of give you an overview of what each of these genes does. And if you don't know what some of these things mean, that's totally okay. The main takeaway point of this slide is just to tell you that the different genes encode proteins that do very, very different things. So the GRIN1 gene encodes the obligatory subunit, and it contains the glycine and D-serine binding site. So what I mean by obligatory subunit is that this subunit has to be present in all NMDA receptors. An NMDA receptor doesn't form without GRIN1. The GRIN2A confers fast deactivation kinetics to the receptor. It contains an intracellular calcium and calmodulin binding site. So there's a part that sticks out in the inside of the receptor and does all these cool intracellular things. And it also binds glutamate. All of the GRIN2 genes actually bind glutamate. The GRIN2B confers medium deactivation kinetics, and it also has a phenylethanol ethanolamine, never said that word out loud, binding site. The GRIN2C has a lower threshold for magnesium blocks, so that magnesium gating mechanism that we talked about before. It also influences the affinity of different ligands to actually bind to the receptor. And there are regions on the GRIN2C protein that regulate whether the receptor will actually go to the surface of the cell or not and act as that channel. GRIN2D binds glutamate and has slower deactivation kinetics than the other GRIN2 genes or proteins. And then GRIN3, the GRIN3 proteins, we know less about. They're the most newly discovered, and we're still kind of figuring out what they do. But we know that GRIN3A proteins form non-conventional NMDA receptors, and it controls the calcium permeability of the channel. And we know that GRIN3B controls magnesium channel block resistance. So, you know, I showed this graphic before, and if you Google NMDA receptors, you get a whole plethora of cartoons that tell you what this receptor actually looks like. And it turns out that it's not as simple as this little, you know, oval channel that sits in the membrane. It's actually got all of these like rolling parts, and the rolling parts are actually what pull these little, like, you know, stringy things apart. And if you want the magnesium block to be relieved, it's actually these rolling mechanisms and the pulling apart mechanisms that allow the ions into the channel. It gets really complicated. But what is important to talk about here is that there are these different regions of the NMDA receptor that are responsible for different things. So this, for example, this ligand binding domain in the middle here, that's what actually binds the D-serine and the glycine and the glutamate. And then the magnesium pore is actually in the transmembrane domain. So it's actually sitting in the middle of the cell membrane. And then the C-terminal domain, like I talked before about with GRIN2A, that's what will have those little signaling mechanisms, those little signaling areas that are responsible for doing cool things inside of the cell. And actually, I'm just going to very quickly make it one step more complicated, just very briefly for illustrative purposes for you all. And that is, second. There we go. Okay. So it turns out that it's actually not a cartoon receptor at all. It actually looks a lot more like this. This is a 3D crystal structure of the NMDA receptor that was first done by Dr. Furukawa, who we'll hear from later. But what I wanted to show you is that, you know, those rolling parts that I showed you in the last screen and those pull apart mechanisms, they're still kind of visible here. So here's that, here's the ligand binding domain of the receptor, and here's the channel pore kind of in the very middle there. I don't know if you can see my mouse, but it's right in the middle there where things kind of get messy. And what I'm hoping you'll see is that there are little different parts that are lighting up. Each of those represents an amino acid. So when I'm scrolling my mouse around and the different parts are turning pink, the different parts of these spirally things are turning pink, that's each indicating a different amino acid. And what the point of me showing that to you is that your child's variant can occur in any one of these amino acids, and we don't know whether you know, it's occurring in a place that's affecting the binding of D-serine or glutamate, or whether it's affecting the magnesium channel blockade. Maybe it's making the magnesium stickier or less sticky, or it may be that the quality control mechanisms that a cell has are stopping the proteins from reaching the channel member, or sorry, the cell membrane at all. And in that case, you know, we're simply having fewer NMDA receptors, period. So just to go back to my my presentation here, I'm almost done. So what's really nice and unique about the NMDA receptor is that it's a really druggable target. Scientists have a big interest in the NMDA receptor because it's so important for how your body functions. And we've developed a lot of different drugs that target it. This is not an exhaustive list. This is just a few, examples that I've decided to show today. But there are like at least 100 drugs that have been developed to target NMDA receptors and that could serve as therapeutic possibilities for the children once we've probed the biophysical properties of how their receptor changes as a result of the mutation. So, D-serine and D-alanine, these are glycine site agonists, and they would be helpful at least for GRIN1 patients. Nevostinel, many words I'm saying today for the first time out loud. This is a positive allosteric modulator at the glycine site for GRIN1. Amantine and ketamine, these are antagonists, so they act inside the channel pore of GRIN1. Ifenprodol, this is an antagonist, and it acts in the N-terminal domain of GRIN2B. Magnesium, so if it's the case where, magnesium just needs more opportunity to bind, magnesium may be a therapeutic possibility for some of the kids. We know that the NMDA receptor has these polyamine and neurosteroid receptor binding sites, so it may be that certain polyamines, certain neurosteroids can help therapeutically. And then, of course, there are drugs that indirectly target the NMDA receptor. So some of the genes that I mentioned, so particularly GRIN-A and GRIN2C, these are transiently expressed during development at higher levels. So what that means is, after your child is born, the gene, the amount of gene tends to change. The amount of protein produced by this gene tends to change. So it may be important for certain parts of development. And in curing those kids or treating those kids, we may be looking at drugs that indirectly target NMDA receptors. So if nothing else, I hope that the key takeaway of this talk is that even though the kids all have different mutations to NMDA receptor genes, they will all manifest in very different ways. And the other big thing that I should mention is that trying an NMDA receptor agonist or antagonist, so something that binds to the receptor or prevents binding to the receptor, it can actually be very dangerous if the science is uncertain about the nature of your kid's mutation. So if you, have an NMDA receptor that's functioning less, and then you give them an antagonist, which prevents it from working even more, your child will probably have even worse seizures. So it can be kind of dangerous, actually, in exploring some of these possibilities. And lastly, the most meaningful way to compare notes among the parents is if parents and caregivers can get together and talk about their children if their children have the exact same mutation. So it's really important to have those meaningful conversations, if you can, and if it's identified, of the exact variant. If there are groups, it's good to talk within those groups about, similarities between the kids and potential therapeutic avenues. So that's all I have. Thank you so much.
Great. Thank you so much to our speakers today and for those wonderful talks. It looks like we have several questions in the chat, so we can take a look at those. A few of the more recent ones look like they'll be more pertinent for Katheron. We have a couple of questions about deactivation kinetics. So one question was, what does fast deactivation kinetics mean? And another question was related to medium deactivation kinetics.
So medium, I just meant that as a sliding scale kind of thing. So, for some of the receptors, if they contain a certain subunit, it happens really quickly, and some of them it doesn't happen as quickly. So what I mean by that is, when the receptor is activated, you've got lots of ions flowing in. So a fast deactivation kinetic means that it would close up very quickly.
Okay, so it looks like we have another question. Anyone out there with GRIN2A missense variation who has good results with any medications? So I'm guessing they're looking for any feedback related to any medications that we know of that work in GRIN2A patients.
GRIN2A?
Yes.
Okay. I honestly don't know. I mean, the most is known about GRIN1 just because it's the obligatory subunit, and we still have to figure out what binding sites are unique to GRIN2A subunits. And from there, we can figure out how to target them with different medications. So none that are specific to GRIN2A yet, as far as I know, but scientists, please feel free to correct me on that.
Perfect. So we have another question. Which method or experiment do you think, oh, let me scrolled away from me there. Which method or experiment do you think is the best and most informative to test the functional effects of a GRIN variant?
Right. So I think that the best way of doing this is to take a mouse and transfect that mouse with the exact variant that's seen in a child, and then to take a brain slice from that mouse and look at the biophysical properties of the receptor. So how is the receptor changed in an organism as a result of those of that variant? And that will give you information potentially about what drugs we should be treating it with. The nice thing about that method is that acutely you can actually just pour drugs on the brain slice and see what happens. And we can get an indicator of whether this would be therapeutically helpful or not. And then from there, those same mice, so their siblings that have that variant, you can test those drugs on them in vivo to see whether that, you know, helps bring their seizures down, helps with their cognitive difficulties, that kind of thing.
Perfect. And we have another very specific question related to that. What is the effect of phenylethylamine binding to the GRIN2B subunit?
I think I'm actually not too sure. I'm not gonna speculate. I'm not too sure, but that is actually an endogenous compound. So that's just what the chemicals that already exist in your body do to the receptor.
Perfect. Okay, so I have another question that might be more applicable for Eriskay or Angie. If we need a test population of 1,000 people for a phase 3 trial, will we ever find enough people? What can be done to make it easier for rare treatments to make it to the market?
Yeah, that's a really great point. So everything will work together. What we need to do is, it's hard. I know it's hard to gather that amount of people. For that reason, the first steps of that cartoon I was showing you to gather information, to create registries and databases is so important. And as a foundation also to help patients to afford the exome sequencing or the clinical tests that are needed in order to gather more patients. So everything needs to move very coordinately. It's not impossible. It's more difficult, but it's completely doable.
Yeah, and I just want to add to that Sick Kids right now is working. One of their main mission and goals right now is establishing sort of a pipeline under the Precision Child Health Initiative to really focus on, you know, the development of trials and therapies for these types of children, and really be able to support that work to be done at a much more ideally fast pace, and setting up the infrastructure. And that's one of the biggest problems, right, is the infrastructure to support these types of you know, therapies and trials that go on. And so Sick Kids is working on that and working to establish a network even across the country, and ideally even more, you know, internationally. It's going to take some time, but that's one of their big visions and goals right now.
Terrific. Thank you for that answer. So it looks like we have another question that might be for Katheron. How does one get a lab to create a mouse of the exact mutation?
That's a good question. There are a few of the labs, including Dr. Collingridge's lab and our lab. We all focus, I think, mostly on GRIN 1, but I'm not too sure who's out there in terms of the GRIN 2 and 3 researchers. It's kind of arbitrarily decide which ones we tackle at a time. So we have 2 mouse lines right now, and I think we have 1 more. And it's really important for us, in the interests of doing a good and thorough job for the patient families, to do them well, kind of one at a time. And, you know, when I think about our lab, you know, we've had 1, being me, and maybe may have another PhD student this year who is actually working on, you know, doing behavioral testing in those animals, and then maybe 1 or 2 people who are doing those biophysical experiments. So it's a slower process. I think I'm not sure whether some mice can be produced and tested commercially. I also know that there are some universities in the US, like the Undiagnosed Health Network affiliated labs, who are dedicated to making these mouse models using CRISPR techniques and then testing them. So I would encourage parents to reach out to labs that do this kind of work, and, you know, and do some patient advocacy and see if you can get somebody to do it. I know that we are full for the next couple years of ones that we'll be able to do, and we wish that we could do more, and we hope to do more. But I'm afraid it's rather arbitrary in terms of whose mouse model gets produced.
Thank you for that answer. So it looks like we have a couple of more mouse-ish related questions. Are there any GRIN to B mouse lines?
There were some. I believe Dr. Katheron Roche, she's at NIH, was developing a couple of them. This would have been several years ago. But I don't know who currently is covering 2B.
Okay. All right, so we have a question that might be for Katheron or Angie. Is it an option to use human brain organoids to test phase 3 treatments instead of patients?
That would be ideal. Thank you so much for that question. It's very interesting. It's not possible. So brain organoids would be in the preclinical stage, even the research and preclinical stage of development. They are a very key research system, as many others, but they are not good for testing in patients, because let's remember that we are a conjunction of a very specific genome background from our mom, from our dad. We have a history as human beings developing, so we need to test in a specific patients with a diverse background too. That's the most important part of the phase 3.
Yep, agreed. Hopefully in future, but not now.
Perfect. Thank you guys for those answers. So we had several questions related to regression. I'm not sure if Eriskay, you might be able to answer some of those. Most of the parents, it looks like they were asking about regression in terms of at different points in life. It looks like some of the parents are describing their child's regression around 3 to 6 months, and they were curious if it depended on the mutation, the type of mutation that their child has.
So I think it's more complicated than that. I think there's certainly what we call some sort of genotype-phenotype correlations, meaning that, you know, you have a specific variant sort of that can sort of predict or at least give a good idea of what the clinical picture will be. But regression, we know can happen, you know, amongst all different types of variants. So I don't think that it's at a point yet where we can really pinpoint which specific variants kids are more prone to have regression. I think it's complex. I think that there's, you know, as, you know, Angie pointed out, you know, we have a diverse, you know, many different genes and many different genes that play a role in brain development and function. And we don't still understand all of the interactions and why some, you know, would experience regression earlier or not at all, for example. So unfortunately, I'm sorry that I'm not able to answer that better, but I don't think we have that answer yet.
Definitely. Thank you for that, Eriskay. So it looks like we have time for maybe one more question or 2 more questions. This one asks, do you have a certain number of children with the same variant before you mimic a mouse with the same deviation? I guess we'll direct this one to Katheron.
So I don't make the rules for how we decide which mice we're going to tackle. I'm not the director of any lab. I'm just a humble PhD student. Ideally, we would. I mean, we get into ethics questions here when we think about it that way. Like, do we decide that someone gets lower priority because they are more like their variant is more likely to occur? Like, how do we determine who is the priority in terms of patient care? We get into some pretty murky ethical questions. And at this time, I don't have a say in how we decide which mice to take on. Yeah.
Great. I think that's probably a good note to end our broadcast on. Thank you all so much for your wonderful talks and for all of the information that you provided us today. And thank you everyone for participating in the Q&A and asking your questions in the chat. For the ones that we have time to get to later, we'll be able to answer those potentially in the Ask Me Anything session at the end of the day. But thank you all so much.
Meagan, can I just say quickly, I did notice in the chat and some of the questions around this question around inheritance and de novo variants and mutations. I'm happy to share with either you or Keith, my contact information. If people want to, I can do, answer those type of questions via email if that would be helpful, because I saw a bit of chat back and forth about that. And if we don't have time to address it now, I'm happy to do it offline via email.
Perfect. Thank you so much for that, Eriskay. Yes, so anybody listening, we're happy to pass on Eriskay's contact information if you have any additional questions. But thank you all so much. And it looks like now we're going to go ahead and take a 15-minute break. Thank you for your participation, and we'll see you in the next session.